how do we develop from single cells into fully functional beings? This long-standing question can be easily investigated with a model organism like our stickleback fish, which have external transparent eggs. So what does direct the development through these stages? We know that genetics are the blueprint for development, and we also know that the timing and location where genes are expressed are crucial to appropriate development. The genes and their timing and location of expression integrate within developmental genetic pathways. The developmental pathway that my lab is interested in is the process of how embryos develop from bipotential cells and then at some point encounter a male or female specific factor, at which point the development of males and females diverge, with each following a separate sex-specific pathway. My lab is especially interested in this point, which we call sex determination. When most people think of what determines sex, they think of X and Y chromosomes. But what many people are surprised to learn is that in genetic sex determination, it is just one single gene on the Y chromosome that determines sex. This gene is known as the master sex determining gene. In humans, the master sex determining gene is a gene called SRY, but there are currently over a dozen known sex determining genes. In our model organism, the three-spined stickleback, AMHY is the master sex determining gene. For my research project, I want to understand how AMHY evolved the ability to work in this developmental pathway. So I first need to characterize when and in what tissue AMHY is expressed. Then, to understand the regulatory pathway that allows for that expression, I need to uncover the pathway of gene regulation leading up to AMHY, which must result in at least one gene eventually turning AMHY on. What are these genes and regulatory elements? My dissertation project seeks to answer these questions. To determine when and where AMHY is being expressed, I am using a technique called in situ hybridization, in which a probe will bind in place to wherever AMHY is being expressed, showing me the location of my gene of interest through the visualization of a color. To determine what genes and regulatory elements regulate the expression of AMHY, I will perform transgenic experiments using various parts of the regulatory region of AMHY to induce sex reversal of XX female fish to XX male fish. I will also perform single-cell sequencing to look deeper into gene expression that may regulate AMHY. With this research, I hope to gain insight into a fundamental developmental pathway and learn about gene regulation during development. This research may be applicable to other areas of development, including when investigating diseases that are caused by a failure of genetic regulatory pathways. Another aspect of this research that I love is its ability to aid in student learning. I taught in high school and college classrooms for a decade before returning to school myself as a PhD student, and during that time I learned that one of the best ways to learn science is through excellent science storytelling. Everyone thinks they have at least some understanding of how males and females differentiate because it is such a fundamental and seemingly simple part of biology. By investigating and skillfully sharing the complexities of sex determination, students both in classrooms and lab can expand their scientific minds and thinking. I look forward to continuing this research and continuing to share this very cool science story. And finally, thank you to the ARCS Foundation for your consideration.